Hello, students. Have you ever noticed how a grass blade feels so different from a sunflower stem? Both are plants, but their inner design, their anatomy, is not the same. Today, we'll step inside the hidden structures of dicotyledonous and monocotyledonous plants. By studying transverse sections, or TS, of roots, stems, and leaves, we see how tissues are arranged in unique patterns. These differences aren't just details for exams, they explain why grasses grow in endless fields and why trees like mango and sunflower grow strong and thick. Let's open the window into the plant's inner world.
Let's begin underground with the dicot root, for example, sunflower. At the outermost layer, we find the epibilma. Some of its cells grow out as fine, unicellular root hairs. These tiny hairs, though invisible to the naked eye, absorb gallons of water and minerals from the soil every day. Moving inward, the cortex is made of parenchyma cells with air spaces for diffusion. The innermost layer, the endodermis, contains Casparian strips, bands of suberin that act like gates, forcing water to move selectively. Just inside lies the pericycle, where lateral roots originate and cambium can arise during secondary growth. The vascular bundles are radial, arranged in alternating patches of xylem and phloem, usually 2 to 4. Together, these tissues form the stele, the central conducting cylinder. Look at this diagram of the sunflower root cross-section. Notice the root hairs, cortex, and radial vascular bundles. Now let's look at a monocote root like MAJ. At first glance, it seems similar. Epidermis, cortex, endodermis, and pericycle are all there. But the vascular system reveals the difference. Monocote roots are polyarch. They have more than 6 xylem bundles. This arrangement provides strong conduction in fibrous root systems. The pith is large and well developed, unlike in dicots where it is small or absent. Most importantly, monocote roots do not undergo secondary growth. That's why grasses remain thin, even as they grow in length. In this TS of MAJ root, notice the many xylem bundles and the large central pith. Moving above ground, let's slice through a dicot stem like sunflower. The epidermis forms the protective outer skin with cuticle and trichomes to reduce water loss. The cortex lies beneath, divided into zones. First, the hypodermis, collenchyma cells that give flexibility. Next, parenchyma with large air spaces. Finally, the endodermis, rich in starch grains, often called the starch sheath. Inside, we see a ring of vascular bundles. Each bundle is conjoint, open, and indarch, with xylem inside and phloem outside. Cambium is present, allowing secondary growth and thickening. At the center lies the pith, a storage area of parenchyma cells. Observe this TS of dicot stem. The ring of vascular bundles is the hallmark of dicots. Now compare that with a monocote stem like MAJ. The hypodermis here is sclerenchymatous, thick-walled and rigid, providing strength. The vascular bundles are scattered throughout the ground tissue, not arranged in a ring. Each bundle is surrounded by a tough sclerenchymatous sheath, and they are closed, no cambium, so no secondary growth. Another difference, phloem parenchyma is absent, and instead, water-containing cavities appear in the bundles. This unique design supports rapid vertical growth of grasses without woody thickening. Look carefully at this MAJ stem cross-section. Scattered bundles make it distinct from the dicot stem. Let's now look at leaves. In dicots, leaves are dorsiventral, meaning the upper and lower surfaces are different. The adaxial, upper, epidermis has a thick cuticle, while the abaxial, lower, epidermis usually has more stomata. The mesophyll is differentiated into palisade cells for photosynthesis and spongy cells for gas exchange. In monocots, leaves are isobilateral, Both surfaces look alike, and stomata are present on both sides. The mesophyll is not divided into palisade and spongy types. Special bulliform cells along the veins help the leaf roll inward during water stress, reducing water loss. Venation also differs. Dicot leaves show reticulate venation, while monocote leaves show parallel venation. In these diagrams of dicot and monocote leaves, compare the mesophyll differentiation and bulliform cells. Let's bring it all together. In roots, dicots show 2 to 4 xylem bundles and secondary growth. Monocots have many xylem bundles, large pith, and no secondary growth. In stems, dicots have a ring of open vascular bundles with cambium. Monocots have scattered, closed bundles with no cambium. In leaves, dicots are dorsiventral with palisade and spongy mesophyll. Monocots are isobilateral with bulliform cells and parallel venation. These differences explain why woody trees and shrubs are dicots, while grasses and cereals are monocots. Isn't it fascinating that the internal design of plants so elegantly matches their life strategies? I hope you now appreciate how anatomy reveals the hidden architecture of plants. From roots in the soil to leaves in the sun, every organ tells a story of adaptation. Biology truly is the science of life in all its detail and beauty. Until next time, keep observing the green world around you with wonder.